Hello, this is Doc Ina and welcome to my lecture on Physiology of Labor. To download my lecture deck, please go to my WordPress site, Docina Obigaine. Reference for this lecture is Williams Obstetrics 25th edition, Chapter 21, Physiology of Labor. And this is the outline of my lecture. So first we review the maternal and fetal compartments. So the myometrium or the myometrial layer of the uterus is composed of bundles of smooth muscle cells surrounded by connective tissue. And the uterine smooth muscle cell is not thermally differentiated and therefore is readily adaptable to environmental changes. There are several smooth muscle qualities that confer advantages for uterine contraction, efficiency, and fetal delivery, and these are the following. So the degree of smooth muscle cell shortening with contractions is greater than in striated muscle cells, and forces can be exerted in smooth muscle cells in multiple directions. Myometrial smooth muscle is not organized in the same manner as skeletal muscle, so the thick and thin filaments are found in long random bundles throughout the cells. And lastly, there's greater multidirectional force generation in the uterine fundus compared with that of the lower uterine segment, which permits versatility in expulsive force directionality. So the decidua is a term we use for the endometrium that is transformed by pregnancy hormones, and this is composed of stromal cells and maternal immune cells. The decidua serves to maintain the pregnancy via a unique immunoregulatory function that suppresses inflammatory signals during gestation. At the end of pregnancy, decidual activation ensues such that the decidua transitions to induce inflammatory signals and withdraw active immunosuppression, which contribute to parturition initiation. During pregnancy, the cervix has multiple functions, and these are the following. So, it maintains the barrier function to protect the reproductive tract from infection. It maintains the cervical competence despite greater gravitational forces as the fetus grows. And it orchestrates extracellular matrix changes that allow progressively greater tissue compliance. In non-pregnant women, the cervix is closed and firm, and its consistency is very similar to that of the nasal cartilage. By the end of pregnancy, the cervix is easily distensible and its consistency is similar to the lips of the oral cavity. So the placenta provides the exchange of nutrients and waste between the mother and the fetus. So the key sources of steroid hormones, growth factors, and other mediators that maintain pregnancy are coming from the placenta, and this potentially aid the transition to parturition. So the fetal membranes, these are the amnion and the chorion, and the adjacent decidua make up an important tissue shell around the fetus that serves as a physiologic, immunological, and metabolic shield to protect against untimely parturition initiation. The amnion provides virtually all of the fetal membrane's tensile strength to resist membrane tearing and rupture. And this avascular tissue right here is highly resistant to penetration by leukocytes, microorganisms, and neoplastic cells. It also constitutes a selective filter to prevent fetal particulate-bound lung and skin secretions from reaching the maternal compartment. Maternal tissues are protected from amniotic fluid constituents that could prematurely accelerate decidual or myometrial activation or could promote adverse events such as amniotic fluid embolism. The chorion, which is right here, is primarily a protective tissue layer and provides immunological acceptance. It is also enriched with enzymes that inactivate uterotonins, which are agents that stimulate contractions. Inactivating enzymes include prostaglandin dehydrogenase, oxytocinase, and enkephalinase. So we now go to sex steroids hormone role. First, estrogen and progesterone. So estrogen promotes and progesterone inhibits the events leading to parturition. Progesterone withdrawal directly precedes progression of parturition. Providing progesterone will delay parturition via a decline in myometrial activity and continued cervical competency. The exact role of estrogen in regulation of human uterine quiescence and cervical competency is less well understood. That said, estrogen can advance progesterone responsiveness, and in doing so, it can promote uterine quiescence. Now, at the end of pregnancy, estrogen aids processes that mediate uterine activation and cervical ripening. Prostaglandins, on the other hand, are lipid molecules with varied hormone-like actions. Now, during parturition, the prostaglandins play a prominent role in myometrial contractility, relaxation, and inflammation. Prostaglandins are produced using plasma membrane-derived arachidonic acid. So, this is arachidonic acid, which usually is released by the action of phospholipase A2 or C. Arachidonic acid can then act as a substrate for both type 1 and type 2 prostaglandin H synthase, which are also called cyclooxygenase 1 and 2 or COX-1 and 2. Both PGHS isoforms convert arachidonic acid to the unstable prostaglandin G2 and then to prostaglandin H2. These enzymes are the target of many nonsteroidal anti-inflammatory drugs or what we call NSAIDs. Through prostaglandin isomerases, prostaglandin H2 is converted to active prostaglandins, and these include prostaglandins E2 or PGE2, F2 alpha or PGF2 alpha, and I2 or PGI2. Another important control point for prostaglandin activity is its metabolism, which most often is through the action of 15-hydroxyprostaglandin dehydrogenase or PGDH. Expression of this enzyme is upregulated during pregnancy in the uterus and cervix, which provides the important ability to rapidly inactivate prostaglandins. Now, the amnion is likely the major source for amniotic fluid prostaglandins, and their role in the activation of cascades that promote membrane rupture is very clear. Now we go to phase 1, which is uterine quiescence and cervical softening. Parturition can be arbitrarily divided into 4 overlapping phases that correspond to the major physiological transitions of the myometrium and cervix during pregnancy. So these phases of parturition include, number 1, a prelude to it, so this one, phase 1, or 2, or phase 2, the preparation for it. Phase 3 is the process itself, or the process of labor, and phase 4 is the recovery. So the phases of parturition should not be confused with the clinical stages of labor, that is the first, second, and the third stages, which make up phase 3 of parturition. So all 3 stages of labor are in phase 3 of parturition. The first stage is divided into a relatively flat latent phase, so this is the latent phase, and a rapidly progressive active phase, this one. In the active phase, there are 3 identifiable parts. So we have the acceleration phase, this part here. We also have the linear phase of maximum slope, this part here, and the deceleration phase. Beginning even before implantation, a remarkably effective period of myometrial quiescence is imposed. This phase 1 normally comprises 95% of pregnancy and is characterized by uterine smooth muscle tranquility with maintenance of cervical structural integrity. All manner of molecular systems, such as neural, endocrine, paracrine, and autocrine, are likely called to implement and coordinate a state of relative uterine unresponsiveness. And moreover, a complementary fail-safe system that protects the uterus against agents that could perturb the tranquility of phase 1 also must be in place. So during phase 1, the myometrial cells undergo a non-contractile state, and uterine muscle is rendered unresponsive to natural stimuli, and this continues until near the end of pregnancy. Now, some low-intensity myometrial contractions are felt during the quiescent phase, but they do not normally cause cervical dilatation, and this is common towards the end of pregnancy. This is what we call the Braxton-Hicks contraction or the false labor contractions. The quiescence of phase 1, this part here, likely stems from, number 1, the actions of estrogen and progesterone via intracellular receptors. Secondly, we have the myometrial cell plasma membrane receptor mediated increases in cyclic adenosine monophosphate or CAMP. Number 3, we have the generation of cyclic guanosine monophosphate or CGMP. And lastly, other systems that includes modification of myometrial cell ion channels. So quiescence is achieved in part by the following. So first we have the diminished intracellular crosstalk and reduced intracellular calcium and ionized calcium levels. So we have ion channel regulation of cell membrane potential, activation of the uterine endoplasmic reticulum stress unfolded protein response, and uterotonin degradation. And contractility, on the other hand, results from the following. So we have enhanced interactions between the actin and myosin proteins, heightened excitability of individual myometrial cells, and promotion of intracellular crosstalk that allows synchronous contractions to develop. So for the actin-myosin interactions, actin and myosin proteins are essential for muscle contraction, and actin must be converted from a globular to a filamentous form. That's a potential mechanism for the maintenance of relaxation is the promotion of actin into a globular form rather than into fibrils, which are required for contraction. So actin must be attached to the cytoskeleton at focal points in the cell membrane to allow tension to develop. And actin also must partner with myosin, which is composed of multiple light and heavy chains. The coupling of myosin and actin activates adenosine triphosphatase or ATPase, which hydrolyzes adenosine triphosphate and generates force. Now, this is catalyzed by the enzyme myosin light chain kinase, which is activated by calcium. Calcium binds to calmodulin, this part here. So, calcium binds to calmodulin, a calcium binding regulatory protein, which in turn binds to and activates myosin light chain kinase. Uterine relaxation ordinarily is promoted by conditions that lower the concentrations of ionized calcium. In contrast, agents that prompt contraction act on myometrial cells to augment calcium or ionized calcium levels, or they allow an influx of extracellular calcium through ligand or voltage regulated calcium channels. Voltage gated ion channels open, additional calcium ions move into the cell, and cellular depolarization follows. For example, prostaglandin F2 alpha and oxytocin bind the respective receptors during labor to open ligand activated calcium channels. Activation of these receptors also increase or releases calcium from the sarcoplasmic reticulum to lower electronegativity within the cell. Additionally, greater localization of non-selective cation channels in the cell membrane promotes calcium entry, and the rise in calcium or ionized calcium levels is also or often transient. But contractions can be prolonged by inhibition of myosin phosphatase, an enzyme which dephosphorylates myosin. Numerous potassium channels also control membrane potential, and one key regulator is the large conductance voltage and calcium activated potassium channel or BK calcium. In normal physiology, the myometrial BK calcium channel plays dual and opposing roles to maintain a balance between uterine quiescence and contractility. Now, for most of pregnancy, opening the BK calcium channel allows potassium to leave the cell to maintain interior electronegativity and thus preventing voltage-gated calcium influx and contraction. Enhancing the BK calcium channel opening results in myometrial relaxation, whereas inhibition of the calcium channel augments myometrial contractility. The ability of the BK calcium channel to regulate calcium dynamics and ultimately uterine contractility from early to late gestation may result from temporal changes in expression of the BK calcium channel and or BK calcium interacting partners. Now, cellular signals that control myometrial contraction and relaxation can be effectively transferred between cells through intercellular junction channels, which you see here. These are the junctional channels. Communication is established between myocytes by gap junction. This is the gap junction, which aid the passage of electrical or ionic coupling currents as well as metabolic coupling. The transmembrane channels, these are the transmembrane channels that make up the gap junctions consist of two protein hemichannels. Now, these connexons are each composed of six connexin subunit proteins, and of these, connexin 43 is expressed in the myometrium and concentrations rise near the labor onset. Pairs of connexons establish a conduit between coupled cells. So this is one connexon here, and this is another connexon here. So a paired connexon there. No? So this establishes a conduit between the coupled cells. So this is one cell membrane, and this is the second cell membrane. And pairing of these two connexons establishes a communication or conduit between these two cells. Now, optimal numbers and types of gap junctions are believed to be important for electrical myometrial synchrony. Progesterone maintains uterine quiescence in part by mechanisms that lower the expression of various key proteins needed for contractility. These contraction-associated proteins, or CAPs, include oxytocin receptor, prostaglandin F receptor, and connexin 43. At the end of pregnancy, increased stretch along with greater estrogen dominance raises CAP levels. Integration of diverse regulatory pathways culminates in released inhibition of connexin 43 and oxytocin receptor levels to promote greater uterine contractility. Progesterone also maintains uterine quiescence through support of myometrial capsase 3, which is an anticontractile agent. This protein degrades both actin and the specific gap junction protein connexin 43. In mice, myometrial capsase 3 activation is regulated by pregnancy-induced endoplasmic reticulum stress response, or ERSR. Functional irregularities cause misfolded proteins to accumulate and trigger the ERSR. The ERSR and its unfolded protein response are cellular mechanisms that work to maintain homeostasis in the face of stimuli such as stress and inflammation. Prolonged ERSR promotes caspase 3 activation to preserve quiescence despite these stimuli. Numerous G protein coupled receptors appear to be modified during the phases of parturition, and several of these are present in myometrium and associated with G alpha's mediated activation of adenylyl cyclase to yield higher CAMP levels. Now, these receptors, the G protein coupled receptors, together with appropriate ligands, may act with sex steroid hormones to maintain uterine quiescence. Examples are the LH receptor and corticotropin releasing hormone receptor 1. Other G protein coupled myometrial receptors instead are associated with G protein mediated activation of phospholipase C, which releases arachidonic acid. And many of these are available to the myometrium during pregnancy in very high concentrations via endocrine or autocrine mechanisms. The beta adrenoreceptors are prototypical examples of CAMP signaling causing myometrium relaxation, and beta adrenergic receptors mediate G alpha stimulated increases in adenylyl cyclase, elevated levels of CAMP, and myometrial cell relaxation. The rate limiting factor is likely the number of receptors expressed at the level of adenylyl cyclase expression. Now, agents binding to these receptors have been used for tocolysis of preterm labor, such as ritodrine and terbutaline. LH and HCG hormones share the same receptor, and this G protein coupled receptor has been found in myometrial smooth muscle and blood vessels. Now, the levels of myometrial LH HCG receptors during pregnancy are greater before than during labor. Chorionic gonadotropin acts to activate adenylyl cyclase by way of a plasma membrane receptor G alpha's link system. This lessens contraction frequency and force and lowers the number of tissue-specific myometrial cell gap junctions. Corticotropin-releasing hormone is synthesized in the placenta and hypothalamus, and CRH plasma levels rise dramatically during the final 6 to 8 weeks of normal pregnancy and are implicated in mechanisms that control the timing of parturition. CRH appears to promote myometrial quiescence during most of pregnancy, but then aids myometrial contractions with parturition onset. In non-laboring myometrium at term, the interaction of CRH with its CRHR1 receptor activates the G-alpha adenylate cyclase CAMP signaling pathway. This results in inhibition of inositol triphosphate and stabilization of ionized calcium levels. However, in term laboring myometrium, ionized calcium concentrations are augmented by CRH activation of G proteins, GQ and GI, and prompt stimulation of IP3 production and greater contractility. Now, for the decidua, now to ensure uterine quiescence, the synthesis in the decidua of prostaglandins, in particular PGF2 alpha, is markedly suppressed. Suppression of prostaglandin production persists throughout most of pregnancy, and suppression withdrawal is a prerequisite for parturition. Phase 1 of parturition also promotes an environment of immune tolerance to protect the fetus, of course. And the decidual stromal cells proactively ensure that fetal antigens do not elicit a maternal immune response, and this stems from a reduced capacity to attract T cells. As for cervical softening, the initial stage of cervical remodeling begins in phase 1 of parturition. It is characterized by greater tissue compliance, yet the cervix remains firm and unyielding. The cervical softening results from increased vascularity, cellular hypertrophy and hyperplasia, and slow progressive compositional and structural changes in the extracellular matrix. Collagen, which is the main structural protein in the cervix, undergoes conformational changes that alter tissue stiffness and flexibility. And specifically, collagen processing in the number of type of stable covalent crosslinks between collagen triple helices is altered. Activity of the cross-link forming enzymes beginning in early pregnancy is also happening. Now we go to phase 2, which is preparation for labor. And this is also what we call uterine awakening or activation, or the progression of uterine changes during the last few weeks of pregnancy. Parturition progression to labor can be blocked by administering progesterone to the mother. Classic progesterone withdrawal resulting from decreased secretion does not occur in human parturition. However, a mechanism for progesterone inactivation, whereby the myometrium and cervix become refractory to progesterone's inhibitory actions, is supported by studies using progesterone receptor antagonists. The diverse mechanisms by which functional progesterone withdrawal or antagonism is achieved may be through the following. So first, we have changes in the relative expression of the nuclear progesterone receptor isoforms PRA, PRB, and PRC. Differential interaction of PRA and PRB with enhancers and inhibitors of gene expression. Alterations in progesterone receptor activity through changes in the expression of co-activators or co-repressors that directly influence receptor function, local inactivation of progesterone by steroid metabolizing enzymes or synthesis of a natural antagonist, and microRNA regulation of progesterone metabolizing enzymes and transcription factors that modulate uterine quiescence. Phase 2 myometrial changes prepare it for labor contractions, and this results from a shift in the expression of key proteins that control uterine quiescence to an expression of contraction-associated proteins. And of these contraction-associated proteins, myometrial oxytocin receptors and gap junction proteins such as connexin 43 markedly rise in number. Now, these CAPs or contraction-associated proteins increase uterine irritability and responsiveness to uterotonins. Another critical change during phase 2 is formation of the lower uterine segment from the isthmus, such that the fetal head often descends to or even through the pelvic inlet is so-called lightening. The abdomen commonly undergoes a shape change and sometimes described by women as baby dropping or baby drop. Myometrial oxytocin receptor levels rise during phase 2 of parturition, and the level of oxytocin receptor mRNA in human myometrium at term is greater than that found in a preterm myometrium. Progesterone and estradiol appear to be the primary regulators of oxytocin receptor expression, And estradiol treatment in vivo or in myometrial explants raises myometrial oxytocin receptor concentrations. Progesterone also may act within the myometrial cell to enhance oxytocin receptor degradation and inhibit oxytocin activation of its receptor at the cell surface. Before contractions begin, the cervix must undergo extensive remodeling, and this eventually leads to the cervix yielding and dilating from forceful uterine contractions. So, the cervical modifications during phase 2 principally involve connective tissue changes, and this is collectively known as cervical ripening. The transition from the softening out to the ripening phase begins weeks or days before labor. During this transformation, the cervical matrix changes its total amounts of glycosaminoglycans, which are large linear polysaccharides, and proteoglycans, which are proteins bound to these glycosaminoglycans. So, for the cervical ripening, we talk about the cervical connective tissues. First, we talk about collagen. So, the cervix is an extracellular matrix-rich tissue, and the constituents of the matrix include type 1, 3, and 4 collagen, matricellular proteins, glycosaminoglycans, proteoglycans, and elastic fibers. Collagen is largely responsible for the structural disposition of the cervix, and this is collagen here. High turnover of collagen during pregnancy likely allows the gradual replacement of mature cross-linked collagen fibrils with poorly cross-linked fibrils, which yield greater collagen disorganization. As for the glycosaminoglycans and proteoglycans, hyaluronan is a high molecular weight polysaccharide that functions alone, whereas most other glycosaminoglycans complex with proteins to form proteoglycans. Hyaluronan is a hydrophilic space-filling molecule, and thus greater hyaluronan production during cervical ripening is thought to increase viscoelasticity, hydration, and matrix disorganization. Hyaluronan synthesis is carried out by hyaluronan synthase isoenzymes, and expression of these enzymes is elevated in the cervix during ripening. Changes in proteoglycan composition are also suggested to accompany cervical ripening. Now, for the uterine stretch, fetal growth is an important component in uterine activation in phase 2 of parturition. With uterine activation, uterine stretch is required for induction of specific CAPs or contraction-associated proteins. Stretch or the uterine stretch increases expression of connexin 43 and oxytocin receptors. Levels of gastrin-releasing peptide, a stimulatory agonist for smooth muscle, are also augmented by stretch in the myometrium. Now, due to overstretching, like for example in cases of polyhydramnios or multifetal pregnancies, they carry a much greater risk for preterm labor than in cases of singleton or normohydramnios. Now, the human fetal hypothalamic pituitary adrenal placental axis is considered a critical component of normal parturition, and moreover, premature activation of this axis, which is the fetal hypothalamic pituitary adrenal placental axis, is considered to prompt many cases of preterm labor. Steroid products of the human fetal adrenal gland are believed to have effects on the placenta and membranes that eventually transform the myometrium from a quiescent to a contractile state. Now, a key component in the human may be the unique ability of the placenta to produce large amounts of CRH. A CRH hormone that is identical to maternal and fetal hypothalamic CRH is synthesized by the placenta in relatively large amounts. However, unlike hypothalamic CRH, which is under the glucocorticoid negative feedback, cortisol instead stimulates placental CRH production. This ability makes it possible to create a feed-forward endocrine cascade that does not end until delivery. In the last 12 weeks of pregnancy, CRH plasma levels rise exponentially or peak during labor and then fall precipitously after delivery. And pregnancies in which the fetus can be considered stressed from various complications, concentrations of CRH in fetal plasma, amniotic fluid, and maternal plasma are greater than those seen in normal. So the placenta is the likely source of this elevated CRH concentration. Now we go to phase 3, which is the labor itself. Labor initiation is heralded by spontaneous release of a small amount of blood-tinged mucus from the vagina, and this extrusion of the mucus or the mucus plug that had previously filled the cervical canal during pregnancy is what we refer to as the bloody show or the show. Now, the passage of the show indicates that labor is already in progress or likely will ensue in hours or several days. Contractions of uterine smooth muscle during labor are painful, and this is because of the following. First is hypoxia of the contracted myometrium, such as that with angina pectoris. Second is the compression of the nerve ganglia in the cervix and lower uterus by contracted interlocking muscle bundles. Third is the cervical stretching during dilatation. And lastly, the stretching of the peritoneum overlying the fundus. Now, uterine contractions are involuntary and for the most part independent of extrauterine control. Neural blockade from epidural analgesia does not diminish their frequency or intensity, and myometrial contractions in paraplegic women and in women after bilateral lumbar sympathectomy are normal, although they're painless. Mechanical stretching of the cervix enhances uterine activity, and this phenomenon is called the Ferguson reflex. Manipulation of the cervix and stripping of the fetal membranes is associated with the rise in blood levels of prostaglandin F2 alpha metabolites. Okay, so the interval between contractions narrows gradually from approximately 10 minutes at the onset of the first stage of labor to as little as every 1 minute or less during the second stage. Periods of relaxation between contractions, however, is very important for fetal well-being or for fetal welfare. Unremitting contractions compromise uteroplacental blood flow sufficiently, and that can cause fetal hypoxemia. So if there's fetal hypoxemia, that could lead to fetal death. In active phase labor, the duration of each contraction ranges from about 30 to 90 seconds and averages 1 minute. Contraction intensity varies appreciably during normal labor, and amniotic fluid pressures generated by contractions during spontaneous labor average about 40 millimeter Hg in pressure, but it can vary from anywhere between 20 to 60 millimeter Hg. Now, the upper segment is firm, or the upper segment of the uterus. This is the upper segment. This is firm during contractions, whereas the lower segment is softer and distended and more passive. This mechanism is imperative because if the entire myometrium, including the lower uterine segment and cervix, were to contract simultaneously and with equal intensity, then the net expulsive force would markedly decline. So the differential in pressure between the upper and the lower uterine segment is very important for us to have that net expulsive force or pressure. Now, the upper segment contracts, okay, and retracts and expels the fetus. Okay, in response to these contractions, the lower uterine segment and the cervix dilates and thereby form a greatly expanded, thinned out tube throughout the or through which the fetus can pass. So from here, no, we have this one where you have a closed cervix, and then because of the contractions of the upper segment of the uterus, that leads to thinning, progressive thinning of the lower uterine segment, effacement, and dilatation of the cervix, and through which the fetus can pass. The myometrium of the upper segment does not relax to its original length after contractions, and instead, it becomes relatively fixed at a shorter length. The upper active uterine segment contracts down on its diminishing contents, but the myometrial tension remains constant. And concurrently, the uterine musculature is kept in firm contact with the uterine contents. As a consequence of retraction, each successive contraction commences where its predecessor left off. So, the upper part of the uterine cavity becomes slightly smaller with each successive contraction. And because of the successive shortening of the muscle fibers, the upper active segment of the uterus becomes progressively thickened throughout the first and second stage. And this process continues and results in a tremendously thickened upper uterine segment and immediately after delivery. By comparison, the lower segment, which is this one, this part here, this is the lower segment, uh, successive lengthening of the fibers with labor is accompanied by thinning, or normally to only a few millimeters in the thinnest part. As a result of the lower segment thinning and concomitant upper segment thinning, so you can see here, this is pronounced in this picture. So you have the lower segment thinning and the upper segment thinning. So there's a boundary between the two, marked by a ridge in the inner uterine surface, in which we call the physiologic retraction ring. Okay, so this is the physiologic retraction ring. This part here is a physiologic retraction ring. Okay, that serves as the boundary between the upper or thicker upper uterine segment and the thinner lower uterine segment. Now, when the thinning of the lower uterine segment becomes extreme, like in this picture here, as in cases of dystocia or obstructed labor, then the ring becomes prominent. Okay, and the ring is what we call the pathological retraction ring of Bandel, or the Bandel's ring. Okay, when you see that pathological retraction ring, that's an ominous sign. Okay, so you have to act fast and probably deliver the baby via cesarean section. Okay, each contraction gradually elongates the ovoid uterine shape and thereby narrows the horizontal diameter, which has important effects on the labor process. Now, there is greater fetal axis pressure. That is, no, the smaller horizontal diameter serves to straighten the fetal vertebral column, and this presses the upper pole of the fetus firmly against the fundus, whereas the lower pole is thrust farther downward. When lengthening of the uterus, the longitudinal muscle fibers are drawn taut. The lower segment and cervix are the only parts of the uterus that are flexible, and these are pulled upward and around the lower pole of the fetus. After the cervix is fully dilated, the most important force in fetal expulsion is reduced by the maternal intra-abdominal pressure. Contraction of the abdominal muscles simultaneously with forced respiratory efforts with the glottis closed is referred to as pushing. Okay, so the force is similar to that with defecation, but the intensity usually is so much greater. Pushing accomplishes little during the first stage. That's why we don't tell our patients to push during the first stage of labor. This will only unnecessarily exhaust the mother. It will just tire her out. And its associated elevated intrauterine pressures may be also harmful to the fetus. For an average-sized fetal head to pass through the cervix, its canal must dilate to a diameter of approximately 10 centimeters. Now, when the cervix dilates to full dilatation, it means that it has reached 10 centimeters dilatation. Cervical effacement is obliteration, or the taking up of the cervix, or the shortening of the cervix, as you can see here. No, so we started with the cervix that is quite long. That's about 3.5 in average length. No, so you can see here that it progressively shortens, right, until it becomes so thin. So that is what you call the process of cervical effacement, or obliteration, or taking up. It is manifest clinically by shortening of the cervical canal from a length of about 3 to 3.5 centimeters to a mere circular orifice with almost paper thin edges, like so. The muscular fibers at the level of the internal cervical os are pulled upward, you can see here, or taken up into the lower uterine segment. And the condition of the external os remains temporarily unchanged. The process of cervical effacement and dilatation causes formation of the forebag of the amniotic fluid. This is the forebag. Okay. This is the forebag here. This is the forebag there. This is the leading portion of fluid and amniotic sac located in front of the presenting part. In the absence of intact membranes, the pressure of the presenting fetal part against the cervix and the lower uterine segment is similarly effective. Early rupture of membranes does not retard cervical dilatation so long as the presenting fetal part is positioned to exert pressure against the cervix and the lower uterine segment. Now, for the second stage of labor, this is where fetal descent happens. In many nulliparas, engagement of the head is accomplished before labor begins. In the descent pattern of normal labor, a typical hyperbolic curve is formed when the station of the fetal head is plotted as a function of labor duration. So this is the descent curve. Okay, it's a hyperbolic curve, no, that is formed by the fetal descent. Station is the term that we use to describe descent of the fetal biparietal diameter or the fetal presenting part in relation to a line drawn between the maternal ischial spines. So station 0 means that the fetal biparietal diameter has passed the level of the maternal ischial spines. Active descent usually takes place after dilatation has progressed for some time, and during the second stage of labor, the speed of descent is maximal and is maintained until the presenting part of the fetus reaches the perineal floor. The birth canal is supported and functionally closed by the pelvic floor, and the most important component of the floor is the levator ani muscle and the fibromuscular connective tissue that covers its upper and lower surfaces. The levator ani muscle closes the lower end of the pelvic cavity as a diaphragm, and thereby a concave upper and a convex lower surface are presented. The posterior and lateral portions of the pelvic floor, which are not spanned by the levator ani muscle, are occupied bilaterally by the piriformis and the coccygeus muscles. In the first stage of labor, the intact membranes and the fetal presenting part serve to dilate the upper vagina, and the most marked change consists of stretching levator ani muscle fibers. This is accompanied by thinning of the central portion of the perineum, which becomes transformed from a wedge-shaped 5 cm thick tissue mass to a very thin, almost transparent membranous structure less than 1 cm thick. When the perineum is distended maximally, the anus becomes markedly dilated and presents an opening that varies from 2 to 3 centimeters in diameter and through which the anterior wall of the rectum bulges. Now we go to third stage or the stage 3 of labor. This stage begins immediately after delivery and involves separation and expulsion of the placenta and its membranes. As the neonate is born, the uterus spontaneously contracts around its diminishing contents, and the uterine fundus now lies below the level of the umbilicus just after the neonate is delivered. This sudden diminution in uterine size is inevitably accompanied by a decrease in the area of the placental implantation site. For the placenta to accommodate itself to this reduced area, it thickens, but because of limited placental elasticity, it is forced to buckle. Okay, so the resulting tension pulls the weakest layer, decidua spongosa, from that site. Okay, so the placental separation follows the disproportion created between the relatively unchanged placental size and the reduced implantation site size. Cleavage of the placenta is aided greatly by the loose structure of the spongy decidua. So, as detachment proceeds, a hematoma, such as in this picture, forms between the separating placenta and the adjacent decidua, which remains attached to the myometrium. The hematoma is usually the result rather than the cause of the separation because in some cases bleeding is negligible. The great decline in uterine cavity surface area simultaneously throws the fetal membranes, that's the amniocorion and the parietal decidua, into innumerable folds, such as this one. Okay, it becomes folded upon itself. And these are then peeled off the uterine wall, partly by a further contraction of the myometrium and partly by traction that is exerted by the separated placenta. After the placenta has detached, it can be expelled by increased intra-abdominal pressure. And completion of the third stage is also accomplished by alternately compressing and elevating the fundus while exerting minimal traction on the umbilical cord. The retroplacental hematoma either follows the placenta or is found within the inverted sac formed by the membranes, and that is what we call the Schultz mechanism of placental expulsion, where blood from the placental site pours into the membrane sac and does not escape externally until after extrusion of the placenta. The other form of placental extrusion is known as the Duncan mechanism. The placenta separates first at the periphery, and the blood collects between the membranes and the uterine wall and escapes from the vagina. The placenta descends sideways, and its maternal surface appears first. Now we talk about the uterotonins during parturition in phase 3. So first, let's talk about oxytocin. During latent pregnancy, during the phase 2 of parturition, the number of myometrial oxytocin receptors grow appreciably. And this increase coincides with a greater uterine contractile responsiveness to oxytocin. Oxytocin is stored and released by the pituitary or the posterior pituitary gland. And maternal serum oxytocin levels are elevated during second stage of labor, which is the end of phase 3 of parturition, in the early Perperium, and during breastfeeding. Now, immediately after the delivery of the fetus, placenta, and the membranes, which completes the parturition phase 3, firm and persistent uterine contractions are essential to prevent postpartum hemorrhage, and oxytocin likely causes persistent contractions. During labor, prostaglandin production within the myometrium and decidua is an efficient mechanism of activating contractions. The fetal membranes and placenta also produce prostaglandins, primarily PGE2 but also PGF2 alpha are detected in amniotic fluid at all gestational ages. Prostaglandin levels in the amniotic fluid are at its highest after labor begins, and these higher levels likely result as the cervix dilates and exposes decidual tissue. These higher levels in the forebag compared with those in the upper compartment are believed to follow an inflammatory response that signals the events leading to active labor. Together, the rise in cytokine and prostaglandin concentrations further degrade the extracellular matrix, thus weakening fetal membranes. Now, for endothelin 1, this is a family of 21 amino acid peptides that powerfully induce myometrial contraction. The endothelin A receptor is preferentially expressed in smooth muscle and when activated, it affects a rise in intracellular calcium. Endothelin 1 is produced in myometrium of term gestations and is able to induce synthesis of other contractile mediators such as prostaglandins and inflammatory mediators. For angiotensin II, 2G protein-linked angiotensin II receptors are expressed in the uterus. That's AT1 and AT2. In non-pregnant women, the AT2 receptor predominates, but the AT1 receptor is preferentially expressed in gravidas. Angiotensin II binding to the plasma membrane receptor evokes contraction. During pregnancy, the vascular smooth muscle that expresses the AT2 receptor is refractory to the pressure effects of infused angiotensin II. Now, lastly, we have the Perperium, which is phase 4. Immediately and for about an hour after delivery, the myometrium remains persistently contracted. Now, this directly compresses the large uterine vessels and allows thrombosis of their lumens to prevent hemorrhage. This is typically augmented by endogenous and pharmacological uterotonic agents such as oxytocin. Uterine involution and cervical repair are prompt remodeling processes that restore these organs to the non-pregnant state. These protect the reproductive tract from invasion by commensal microorganisms and restore endometrial responsiveness to normal hormonal cyclicity. During the early Perperium, lactogenesis and milk letdown begin in the mammary glands, and reinstitution of ovulation signals preparation for the next pregnancy. Ovulation generally occurs within 4 to 6 weeks after birth. However, it is dependent on the duration of breastfeeding and lactation-induced prolactin-mediated anovulation and amenorrhea. So that's it for my lecture. So in summary, we talked about or reviewed maternal and fetal compartments and the sex steroid hormone roles and the prostaglandin roles in labor. So we also talked about phase 1, phase 2, phase 3, and phase 4 of labor physiology. So that's it for my lecture. Thank you for watching this lecture, and please don't forget to subscribe in my YouTube channel and my WordPress site, Doc Ina Obigaine. Thank you.